Hi everyone, here is a brief introduction of my abstract named Postnatal appearance promotes the thickening but not myelination of the neonatal ventral visual cortex. My name is Mingyang Li, a postdoctor from Zhejiang University. My present work is to answer a question that's whether any postnatal appearance influences the structure development in human visual cortex. We used the public data from Developing Human Connectome Project, the DHCP. After data filter, there are 407 subjects in this analysis. The right figure shows the distribution of the postmenstrual age, namely PMA of the neonate, in our study. The distribution is nearly norm, and most babies were born between 14 and 42 weeks. First, we define the anatomic mask of ventral cortex using HCP-MMP atlas in adult space. The mask contains 34 regions of interest per hemisphere and mainly include the early visual cortex, ventral occipital temporal cortex, and anterior inferior temporal area. We register this mask into neonatal space by a surface-based registration method called multimodal surface matching. The resulting mask was visually checked to assure that the anatomic location is approximately right. As shown in the left figure, the area V1 basically covered the Kerckerian circles in adult space, and you can also see that in the neonatal space. And the VOTC, the ventral occipital temporal area, covered the occipital temporal circles and the collateral circles, and it also can be observed in the neonatal space. Then we extracted the cortical thickness and the cortical myelination in the whole mask and applied the correlation analysis between the structure measurements and the PMA. In general, the mean cortical thickness and the myelination of the ventral cortex significantly improves between 13.7 to 14.5 weeks of PMA, as shown in the right figure, and the blue indicates the left hemisphere and the pink is the right hemisphere. Then we did the correlation analysis in each of the 34 LIs in each hemisphere to see the spatial difference of the development. As shown in the figure A and the right figure, both cortical thickness and myelination show high correlation with PMA in the posterior than anterior region, and high correlation in the medial than lateral part within the anatomic mask. This result is consistent to the previous study, but what they didn't do is to separate the effect of postnatal time in the PMA of neonates. So when we did the correlation analysis between the structure measurements and the postnatal time, we found that the cortical thickness significantly increased with the postnatal time, but the cortical myelination was not. Furthermore, we applied the partial correlation to control the PMA in the above analysis, and the effect of cortical thickness was still significant. In addition, the correlation between cortical myelination and the postnatal time was significantly negative in all LIs after controlling for the PMA. This result might reflect the impact of prenatal time on the myelination, because subjects who have a longer postnatal time would have lower prenatal time at birth with a given PMA. We further validated our results in two subsets. The first subgroup contained neonates whose postnatal time was less than 3 days, which means the subjects in this group almost have no postnatal appearance. The second subgroup contained neonates whose PMA will range from 14 to 42 weeks to control the PMA but keep the variation of postnatal time in this group. So, if the postnatal time do have modulated effects on the development of cortical thickness, the correlation in the first group should be weaker or no more significant. but the correlation in the second group should still significant. And we see in the first subsample, the cortical myelination shows strong correlation with PMA, but the cortical thickness was not. And in the second subgroup, significantly positive correlation remained between cortical thickness and postnatal time in ventral cortex, and significantly negative correlation was found between cortical myelination and postnatal time. These results supported above findings that the development of cortical myelination depends on the endogenous mechanisms, and the longer prenatal time would be beneficial for the development of cortical myelination. In contrast, the development of cortical thickness in neonates was driven by both postnatal time and endogenous growth. Relative to term-born infants, preterm-born infants have longer postnatal time, with relatively immature development at birth, and thus the comparison between them may help us to understand the influence of early experience and innate immaturity on cortical development. And 14 sex LIs show significantly low cortical thickness in term-born than preterm-born infants, and all LIs show significantly high cortical myelination in the term-born than preterm-born infants. This result shows that the early development of cortical thickness in neonates could be modulated by postnatal time even before the term-born time, which is commonly 37 weeks. In conclusion, our study suggests that the development of cortical myelination in neonatal period probably depends on the endogenous mechanisms, but the development of cortical thickness in neonates was driven by both postnatal time and endogenous growth, and the postnatal appearance could accelerate the thickening of neonatal visual cortex, even if it was not necessary. In the end, and thanks to the DHCP for the precious data they shared, and the guidance from the corresponding author Dan Wu, and the suggestions from the people in our lab. That's all, thank you.